if you just kind of want to make sure you're muted so that there's no kind of feedback and things like that. So macromolecules. So hopefully in your 1080 section, you have started to learn about carbohydrates, lipids, proteins, and nucleic acids. Could you really quickly use the raise your hand feature to tell me if you have started learning about the macromolecules in lecture? Awesome. So I see Adrian has, Christina, great, Jasmine. Awesome. So it looks like a lot of us have. If you haven't started learning about the macromolecules yet, you will soon. I promise it's gonna, every section of 1080 does start talking about it at some point. But really the macromolecules, these 4 classes of macromolecules, are really required for many of the building blocks that make things like our cells, the organelles that we find inside of our cells, the tiny little organs, our tissues, our organs, our bones, like really anything that you can think of in our body, there is most likely a macromolecule kind of working in the background to provide that structure. The term macro means large. So a macromolecule is really just any large molecule. But there are 4 classes that are highlighted in red here on this slide that are very much required for life. And those are the carbohydrates, examples of which include like glucose, which is a simple sugar, or starch, which is a more complex sugar. Nucleic acids like DNA and RNA. We've heard a lot about RNA with the COVID-19 vaccines. Proteins, we're going to see proteins are really the most diverse class of macromolecule, can range from anything like insulin, which helps regulate your blood sugar. If you just ate breakfast, your body is probably, your pancreas specifically, is probably releasing insulin to help regulate and lower the blood sugar from your breakfast. Venom, so when an insect or a poisonous reptile bites you, venom is actually made from protein. And hemoglobin, which carries our oxygen in our blood, is also a protein. And then the last category of macromolecules are the lipids, which make up the bulk of our cell membrane that forms the boundary between our cell and the external environment of the cell, and fats, like things like olive oil, cooking oil, things that we kind of experience day to day if we cook, are also in the lipid category. So because so many of you raised your hands to say that you are indeed learning about the macromolecules in class, I am going to kind of keep my overview as simple as, or kind of as big picture as possible, just because we're all a little bit in a different spot, but this lab should be review for most of us in terms of the kind of intro information that we're learning. The one thing that I want to point out about all of the macromolecules is the fact that another term that we can use for these, sorry, my pen is being funky, is polymer. Poly means many, mer means unit. So polymers are many units that are stuck together. And polymers specifically are made from what are called monomers. Mono meaning singular, mer meaning unit. So if you look at this table, and I apologize, it's a little small, but all of our 4 classes, carbohydrates, nucleic acids, proteins, lipids, all of those macromolecule classes are made from smaller building blocks, from monomers. For instance, carbohydrates are made from monosaccharides, or single sugars. Saccharide is just another term for sugar. Nucleic acids are made from nucleotides. Proteins, their building blocks are amino acids. And lipids are a little bit diverse in what can build a lipid, but one of the main components of all lipids, one of the main building blocks, are the fatty acids. So I've just rewritten those names of the monomers in case that figure is a little blurry or small for you. But those are important to keep in mind. we can't make these large macromolecules unless we link these smaller monomers together. So we have to make these mini unit complexes from individual units. And each class has a different unit or building block that makes that up. So what I want to do kind of quickly now is just go through these 4 categories of macromolecules just so that you guys can kind of see what they look like. We're going to talk just quickly about some key differences and functions between these different macromolecules, because today in the lab, you're going to see that we can actually use color indicators similar to what we did for pH. We can use indicators that change color that allow us to test for the presence of these different macromolecules. So instead of testing for pH this week, we're going to be testing for the presence or the absence of a macromolecule, but we're going to be using a color indicator to do that. So just to kind of quickly go over like things like carbs, proteins, lipids, and nucleic acids. For the carbohydrates, we will talk a lot, really, both in lecture and lab, about glucose, which really is considered one of the main building blocks of all of our other complex carbohydrates. So glucose is a monosaccharide, C6H12O6 is its molecular formula. And most complex carbohydrates are a derivative of glucose. There are other monosaccharides as well, like there's fructose as another example. But we can see that both, if we look at the molecular formula for both glucose and cellulose, notice that there's really only 3 elements that make up these molecules. It's carbon, hydrogen, and oxygen. And they typically are found in a 1:2:1 ratio for the monosaccharides specifically. That ratio can kind of change as we start linking those monosaccharides together. But carbohydrates, we can either be very simple, those monosaccharides, those building blocks, or we can link monosaccharides together to create, say, something like cellulose. Cellulose is the main component of the cell wall of plants. It's what allows that cell wall to be rigid and stiff. The way that we link monosaccharides together, and really any monomer to create a polymer, is through a reaction called a dehydration synthesis reaction, or just a dehydration reaction. Anytime we want to make a polymer, again, because we have to link monomers together, we have to perform a chemical reaction to do that. And specifically, what we're going to do is we are going to remove water. So for example, in this reaction here on the bottom, on the left-hand side, we have 2 reactants, glucose and fructose. Both of these are monosaccharides, and highlighted in blue are the 2 hydrogens and 1 oxygen that will be removed from both of these molecules. Together that makes water, so we can see that water leaves our reaction. And by dehydrating, by removing water from our monosaccharides, we can link those together to create a molecule of sucrose. Sucrose is a disaccharide, di meaning 2 sugars are attached together here. And we commonly know of sucrose as table sugar. This is the sugar that you use to bake, or if you add it to your coffee or tea in the morning or afternoon, or whenever you need a sweet pick-me-up. So sucrose is an example of how we can link our monomers together. And every time we do that, we're going to dehydrate our molecules. We're going to remove water. And we can perform a series of dehydration reactions to create something like starch, which is a complex carbohydrate, which typically has hundreds, if not thousands, of monosaccharides linked together. So kind of the structure of starch is shown here on the right. This bracket with the letter N here just denotes that this can kind of continue. This is the repeating unit that makes starch. So starch can be really, really long and really complex. Again, starch is specific to plants, and starch acts as basically energy storage for the plant. So when plants perform photosynthesis, they make a ton of glucose. and they need to store that for later use, and so it gets stored in the form of starch. Animals have something similar. We have a complex carbohydrate called glycogen. Right now, if you just ate breakfast, your liver is actually working to make glycogen. This is how we store kind of our carbohydrate reserves for later. When our blood sugar drops, our liver can be signaled to, specifically the pancreas releases a hormone that signals to our liver and says, hey, I need sugar, can you start kind of breaking down your glycogen and releasing glucose into the blood so that I can get my blood sugar back to normal. So we also have a form of starch, it's just different, it's called glycogen. So we can have simple sugars, or monosaccharides. We can have kind of medium of the line simple, where we link just two saccharides, monosaccharides together to create something like sucrose. Or we can link hundreds, if not thousands, of monosaccharides to make things like starch and glycogen. But all of these are functioning really in energy storage. You'll talk a lot in lecture about how glucose particularly can be used to make ATP, which is our kind of usable energy that our cells use in the body. Nucleic acids, we're going to talk a lot about these later in the semester when we get into lab 14. So for right now, I'm just going to keep this pretty simple. Again, the building blocks for nucleic acids are nucleotides, and there are 5 nucleotides, but they are specific to whether we are making DNA or RNA. The building blocks for DNA are adenine or A, thymine T, cytosine C, or guanine G. For RNA, we also use adenine, but instead of thymine, we use uracil or U. And we also can use cytosine and guanine. So depending on which nucleic acid we're trying to make, DNA or RNA, we're going to use different building blocks. But overall, the function of nucleic acids is ultimately to store genetic information. So our DNA houses our genes. This is basically kind of like the library of information that makes us as human. And then RNA kind of works as a middleman. It basically it's like photocopying like a chapter from, like, say, an encyclopedia, and it just takes like a subset of that encyclopedia, and then it can be used to make things like protein in the cell. And then proteins, again, are really our most diverse class of macromolecules. They are all categorized on the fact that they are considered nitrogenous, meaning they contain the element nitrogen. And again, the building blocks of proteins are the amino acids. Hey, Cassandra, it looks like your camera might have unmuted. It's fine if it's, I don't care if it's fine with you. I just wanted to let you know, just in case you didn't want that unmuted. Proteins are really essential to all of life. Really, any function that you can think of that needs to be performed in our body is probably performed by a protein. So even things like your hair color, your skin color are controlled by proteins. A lot of our proteins are enzymes, which means they actually help ensure that chemical reactions happen in a kind of realistic time span within our body. We have proteins that work to keep our tissues kind of together and organized. Our antibodies that work in our immune system to help us prevent infection or getting sick from pathogens are also proteins. So really, really diverse class, but all of these are made from amino acids. The amino acids make up a family. There are 20 different building blocks or amino acids that make up proteins, and those are shown in this figure here on the right-hand side. Again, I know the figure is somewhat small. I'm never going to require that you know all of the amino acids and their structure, but in general, know that there are 20 amino acids, and they can be categorized depending on how they behave chemically. When we want to create a protein, which is often referred to as a polypeptide, So same, polypeptide is just another name for protein. We can also perform a dehydration reaction where we remove water, and that allows us to link two amino acids together, as we see kind of in the left-hand side here. So we remove water from two amino acids, we can link those together. The name of the bond that is created when we link amino acids together is a peptide bond, which is where polypeptide comes from. So many amino acids linked together by many peptide bonds make a polypeptide or protein. Another term I just wanted to bring up, and I'm actually, you can just cross off this information on this slide. It's not necessary for lab, and I'm not going to test you on it, but I did just want to highlight that condensation reaction is another term for dehydration reaction. So just kind of as if you come across that in lab or your textbook or in class, know that these two things are synonymous. In both cases, you are removing water in order to link our monomers together. But the information that's here, just cross that off. It's really not pertinent to our lab today, so I don't want to kind of bother you with that. Also, just for time's sake, I'm going to skip kind of talking about the structure of proteins. You're going to see in lecture that proteins there are 4 levels which ultimately lead to kind of the final protein structure. This is a nice figure that you can definitely use to help you study for that, but again, it's not necessary to this lab, so I'm just gonna pass over it, but I didn't want to delete it. I wanted to just tell you that you don't need to know for lab. And then again, proteins, this picture of hemoglobin is just highlighting that different proteins have different shapes, and it's really the shape of the protein that determines how it functions. Hemoglobin, which carries, again, our oxygen in our blood, will always have this specific shape. There's a molecule or an atom of iron, or Fe, that sits kind of in the middle of this pocket, and it's that iron that is capable of carrying oxygen gas. So kind of basically, hemoglobin kind of makes like this inner tube that iron can sit inside to carry oxygen. But again, this is just to highlight that proteins have a specific 3D shape. It's really not important to our lab today, so I'm gonna kind of skip over it so that we can get into the lipids. So the last category of macromolecule that you guys are going to be testing for are the lipids. And there are 2 kind of main lipids that we talk about in at least Bio 1080 and 90. And the first type of lipid are the phospholipids. The phospholipids are really important because they make up the majority of our cell membrane. I'll show you how they arrange themselves to do that here in a second. All lipids are categorized as being a lipid because these are very hydrophobic molecules, meaning they hate water. They're very, very afraid of water, and they would like to have nothing to do with that molecule if at all possible. Phospholipids are funky in they actually have a region that is hydrophilic, meaning it loves water, and a region that is hydrophobic that hates water. This kind of top region that I'm circling in red that often gets simplified and drawn just kind of as a circle, this region is often referred to as the phosphate head. This region is very hydrophilic. So the phosphate head of a phospholipid loves water and wants to interact with water. But hanging off of that phosphate head are these 2 fatty acids, which often, again, just get drawn as kind of 2 strings hanging off of our balloon. Those fatty acids are hydrophobic. So these components of our molecule do not want anything to do with water. When something has both hydrophilic and hydrophobic regions, we can call that like amphibian, pathic. So just like an amphibian can go on water and land, amphipathic molecules have regions that love and hate water. And having this amphipathic nature to this molecule is really important because it causes these molecules to arrange themselves in a very specific way when they create our plasma membranes for our cells. And the structure that they create is referred to as the lipid bilayer, which is shown here on this slide. So it's important to remember that our bodies are mostly water. The kind of outside environment of cells is mostly water. The inside of cells is mostly water. And so these phospholipids are going to arrange themselves so that the hydrophilic heads face those watery environments. So we're going to have basically two layers of phospholipids coming together to create a bilayer. And they arrange themselves so that the hydrophilic regions face the water-based environments. So now these portions of our molecule are happy. They get to interact with the thing that they love. By creating a bilayer, what also happens is that it allows these fatty acid tails to be in a region that has absolutely no water. So in the middle of this bilayer, we have just our fatty acid tails. And because there is no water, these molecules are also happy as well. So they get to be in an environment where they don't have to deal with the thing that they have a phobia for. So this is why phospholipids arrange themselves in this very specific way. And when you get into chapter 7 of your textbook, you'll learn more about the plasma membrane in general, but you'll see that the lipid bilayer, these phospholipids really make up the main component of our plasma membrane. You'll see in lecture that the cell membrane itself, there's a lot of stuff kind of shoved into that lipid bilayer, which is just highlighted here in this image. So we also see other macromolecules like carbohydrates and proteins. We see other lipids like steroids also in that lipid bilayer. So while the phospholipids are very much required for making kind of the bilayer portion of our cell membrane, there are other macromolecules that are also key to the proper functioning of the cell. And the last kind of type of lipid, and one that you're going to be interacting with today in lab, are the triglycerides. Triglycerides make up kind of our common cooking materials. So think like olive oil, butter, kind of all of the common like oils or fats that we use for cooking are made up of triglycerides. And triglycerides are very hydrophobic. The entire molecule is hydrophobic. And they get their name because they have a glycerol molecule, so that's the glyceride portion, attached to 3 fatty acids. So the tri in triglyceride is referring to the fact that you have 3 fatty acids hanging off of that glycerol molecule. And you're going to be seeing kind of how we can test for triglycerides specifically in lab today. So kind of with that, for Lab 5 today, you are going to be describing whether solutions have or don't have any or all of these 4 classes of macromolecules. And like I said, similarly to our pH lab last week, you'll be using indicators that change color in the presence or absence of those molecules. In part 1, you're going to watch a video where you know what is in your test tube. And so it's just going to show you how your color indicators should react in the presence of carbs, proteins, lipids. And then in part 2, you're going to watch a video where you have 2 unknown solutions, unknown A and unknown B. And based on how our indicators change color, you'll be able to tell what the composition of macromolecules is in those solutions. And then the last part is we're really just looking at the chemical structures of the molecules by looking at molecular models, which is pretty straightforward. Before I let you guys go, I do just want to quickly let you know how the indicators work that we're going to be using. And you can actually kind of start filling out the table that is in your lab notebook if you'd like to do that as we go through these. But there are different indicators that we use to test for different macromolecules. For example, if we want to test for a complex carbohydrate, we are going to use what is called Lugol's iodine. or just iodine. It's named after the scientist who like developed this. But Lugol's iodine, in the absence of, say, starch, like a complex carbohydrate, Lugol's iodine will look kind of like this gold color here on the right. However, if starch is present, if you have a positive result, that iodine turns really dark purple. It turns almost black. It's so purple. So this is used to test for things like starch. I want to highlight that, remember, starch, our complex carbohydrates, are made from monosaccharides, simple sugars. This solution will not detect those simple sugars. We have to use a different indicator to do that. And that indicator for simple carbohydrates is called Benedict's solution. Benedict's solution is in the absence of sugar, which is shown on the left-hand side of this figure here. It should be blue. If you have glucose or another simple sugar, after you heat it, so Benedict's is kind of funky in that it won't change color unless you boil it for a little bit, but after heating, if there are simple sugars present in your test tube, it should turn kind of a burnt orange color. This third test here on the right is just showing you that if you have sucrose, which remember is not a monosaccharide, it's a disaccharide, it will also stay blue. So Benedict's only sees monosaccharides specifically. And the key thing that you have to do with Benedict's is you have to heat it up before it'll change color. And what's kind of cool about Benedict's is that it can actually kind of give you different qualitative results for different amounts of monosaccharides. So you can actually sometimes get a gradient of colors after you heat Benedict's that allow you to determine if there's a little bit or a lot of simple sugar in your test. So again, Benedict's should be completely blue if there's no monosaccharide, but as you kind of go up in concentration, it'll change anywhere from kind of a green for very low levels of monosaccharide to this really brick red color for very high amounts. In our labs, we're always going to get something near the brick red color because typically you're being given a solution that has a lot of monosaccharide in it. So carbohydrates, we can use either iodine or Benedict's to test for those. If we want to test for proteins, we're going to use what is called biuret A and B. This again tests for polypeptides, not individual amino acids. If there is no polypeptide in your solution, biuret A and B should be this blue color. But if you do have a positive or you do have proteins in your solution, it should turn kind of like a violet blue color. You don't have to heat this up, it'll just turn kind of instantly as if you have that compound or not. So biuret A and B is used for proteins. And then the last indicator that you're going to use tests for a lipid, and that indicator is called Sudan 4. How Sudan 4 works is it stains lipids. So if you had a test tube that had lipids and water, remember lipids are hydrophobic, so they're going to kind of make a separate layer from water. If you've ever mixed oil and water, you've seen this. And so Sudan 4 will specifically stain the kind of layer that contains your fats. So when you have lipids in your solution, you should get a test tube like we see on the left, where you have kind of this separation of clear and dark red. If you don't have lipids in your solution, it should look like the test tube on the right-hand side, where you kind of just get this kind of random disbursement of Sudan 4, and it's not really sticking to anything in your test tube. So those are the 4 indicators that you're going to use. Before I let you guys break out into groups, I just want to remind you that next week we are going to be doing Lab 6, which is on cell structure microscopy. We're going to be using a lot of the same math that we were using in Lab 4. So if you want to kind of review that microscope lab, that can be really helpful before coming to class. And then quiz 4 next week will cover Lab 5 material. But again, the big thing that you need to complete today is the Lab 5 and your Quiz 3. At this point, I am done lecturing, and so I'm going to let you guys break off into your group.